In this video, we're going to talk about the structure of eukaryotic cells. Now, just remember, by definition, eukaryotic cells means that it's going to have a membrane-bound nucleus. This is something we didn't see in our bacterial or prokaryotic cells. In addition, the eukaryotic cells will have other membrane-bound organelles such as the ER, the Golgi apparatus, mitochondria, etc. And that means that the eukaryotic cells are going to be more complex than those prokaryotic cells. And they're not just going to be microscopic. Some of them are going to be multicellular and quite large and macroscopic. The eukaryotic group is going to contain those fungi and the algae, plants, animals, etc. So we're going to go through the various structures. But remember, as with the prokaryotic cells, some of these structures are going to be found in all eukaryotic cells. So they're plasma or cell membrane, cytoplasm, ribosomes, DNA, and then of course they're going to have membrane-bound organelles. Some are not going to be found in all cells, such as not all of them are going to have a cell wall or flagella or cilia. So let's first look at what we refer to as those external structures. So they're on the outside of the cell. And these are going to be for boundary, they're going to be for attachment and motility. So we're going to look at the appendages and the boundary itself. So the appendages are going to be the flagella and the cilia. These are both going to be protein-based structures that we refer to as microtubules. For the flagella, the microtubular arrangement is a 9+2 arrangement because it has 9 on the outside perimeter or circle and 2 on the inside. Because of that structure, the flagella in the eukaryotic cells is going to allow for motility, but that tail is going to go in a back and forth or whip-like motion. Think of how you consider a sperm tail that moves back and forth. This is different than in prokaryotic cells, which they have a flagella in some cells as well, and it does allow for motility, but that flagella will move in a circular motion, not a whip-like motion as within eukaryotic cells. The cilia that is found in eukaryotic cells is going to be small. They're going to be numerous. They may be on one end of polar or they may be around the whole cell, but they have a little different function in the fact that what they're going to do is these cells are often going to be in an aqueous type environment. The cilia is going to move. It moves that fluid around the cell and it allows the cell then to filter and use whatever's out there in its environment for feeding and for nutrients, etc. Now, in eukaryotic cells, we refer to the cell boundary as that structures which then will separate the cell from its external environment. And in eukaryotic cells, the cell boundary may be composed of 2 different parts. All cells are going to have what's referred to as that plasma membrane. The plasma membrane is that very familiar phospholipid bilayer that's going to have embedded proteins and sugars. And because of that embedded proteins and sugars, it's often referred to as the fluid mosaic model, as you can see over here. So that's going to be part of that cell boundary, and all cells are going to have that. Now, some cells will also have a cell wall. Cell wall is going to vary very much on its composition, its structure, based on which group or what the function of that cell wall is for. Now, together, if it has a cell wall and then that plasma membrane, those form what is known as the cell boundary. For relative sake, think about in the prokaryotic cell, we refer to a cell envelope, and it had up to 3 components. So that's a similar structure as what we refer to as the cell boundary in the eukaryotic cell. Now let's start looking to internal structures, and these are going to be your cell organelles. Now, let me just remind you, an organelle just means that it is a structure within the cell that's going to help give it a specific function. Organelles may not have membranes, so some of those membrane lists or without membrane organelles are going to be your ribosome cytoskeletal pieces. Those, remember, were also found in prokaryotic cells. We're going to concentrate here on the membrane-bound organelles, which are unique for eukaryotic cells. And when we talk about membrane-bound organelles, recognize that they can either be single membranes, such as our lysosomes, our Golgi apparatus, ER, or they may be double membraned, such as the nucleus and the mitochondria. So let's first start with the most prominent out of all the organelles and what makes it truly a eukaryotic cell. That is going to be the nucleus. The nucleus is the genetic information of the cell. And we say that because within the nucleus, you're going to find chromatin, chromosomes, and a nucleolus. So when we talk about it, again, it is a double membrane structure. So you see here, and you can see the two membranes and the pores that allows things in and out. And then within that nucleus, you see this very prominent dark purple structure here. That is known as the nucleolus. And the nucleolus is going to have the RNA ability to synthesize RNA there, okay? So that is going to be where you're going to encode for the ribosomal RNA. Then you're also going to find that you're going to have chromatin, okay? And chromatin is going to be those pieces that are going to be made up of DNA in the histones. It's that genetic material, and the chromatin ultimately is going to be twisted and turned into chromosomes. So within the nucleus, remember, it's double membrane. It's got the nucleolus where you're going to do your RNA synthesis, and then it's got the chromatin, which is going to be that DNA and histone proteins. Now, again, here's the nucleus with its double membrane, and coming off of the nucleus, we're still internal, we're still with our organelles that are membrane bound. You'll see here that you have two structures that come straight off, and you see some have the red dots and then some don't. That is referred to the endoplasmic reticulum. And endoplasmic reticulum is used for basically packaging, creating, storage. Okay, so let's look a little bit here at that endoplasmic reticulum. Again, there's two different types. Here where you see the red dots, that is what we refer to as the rough ER or the RER. And the reason it's called rough is because microscopically it looks rough, and that's because on those surfaces are these little red dots known as ribosomes. Now, the rough ER is going to come straight off of that nuclear envelope, so that double membrane part of the nucleus, and it's going to have this very intricate folding network of membrane structures. It's going to allow for what's made in that nucleus, what is going to be needed in the nucleus. It's going to allow for transport material from that nucleus, from the nucleolus in particular, all the way out, potentially even to the exterior of the cells. So in the rough ER, in particular, it has these ribosomes, and the ribosomes are going to be the site for protein synthesis. We're going to learn more about that in our genetics chapter. All you need to know right now is that because of the ribosomes on it, the rough ER, not only is it for transport and storage, but it is also for protein synthesis. Now the smooth ER, as you see here, again called smooth because microscopically you don't have any of those bumps created by the ribosomes, but it is very similar in function in the fact that it's a tubular network. It is going to be for transport, storage, etc., but it's also going to be for synthesis, but not for protein synthesis. It is going to be used for lipid synthesis. So we go from the nucleus here then to the endoplasmic reticulum, which is going to be for synthesis, for transport and storage. And now you'll notice that right off of that you have this structure over here, and again it's a membrane, single membrane bound structure. It looks like folded pancakes, if you will. Each of those is a disc, and each disc is called a cisternae, okay, or cisternae for plural. So unlike the ER, which is a continuous network, each one is its own flattened sac or disc. Okay. Now, what happens here is the ER will have some transport vessels that will come in, and they will be picked up by the Golgi apparatus. Once the materials are within the Golgi apparatus, the Golgi is made for modifying those structures that are made in the ER. So remember, we talked about, let me go back one slide, the ER is for protein synthesis and lipid synthesis. Those components will come out of the ER, they go to the Golgi apparatus, where the Golgi apparatus will then modify things, and then it will move things out of the Golgi, and again it's going to be in a vessel, but that vessel then is, as it pinches off, is going to be a lysosome or another type of vascular, or that is going to then move through the cell, stay in the cell, possibly go outside of the cell, depending on what the component is. Another membrane-bound organelle that is found in the prokaryotic cells is known as the mitochondria. The mitochondria is a double membrane Okay, so it has a double membrane. It's often referred to as the powerhouse or the energy factories, okay, because that's where most of the metabolic respiration occurs for eukaryotic cells. So it's going to be a site for energy production, ATP. And it's going to be able to do that because it has all of this. You can see there's one membrane in the dark purple. The second membrane is folded. It has those folds again are called the cristae, and it's because of that double folded system that allows for ATP production and that electron transport system. Now, just a couple of other internal structures that we're just going to briefly hit on for definition kind of sake is the lysosomes. Now, remember, lysosomes were produced in one way by the Golgi apparatus, and they're going to have various enzymes, which are proteins. They're going to have various different nutrients, protective chemicals, etc. And so they're going to be either contained within the cell, or sometimes, as I mentioned, they will get pushed to the exterior of the cell, especially if it's any kind of debris or damaged material that needs to get rid of. Another vesicle that you will find inside some of the eukaryotic cells is what's known as a peroxisome. Peroxisomes are going to have the enzymes, and they're going to help with detoxification. Okay, one of the chemicals found in there is a peroxide type chemical, and that's part of how we can then break down toxic substances and help the cell get rid of it. Vacuoles are also an internal structure. These are membrane-bound sacs, and they're going to be either fluid-based or solid. They're going to be used for various functions within the cell, such as digestion, excretion. They may just be storage sacs. It's going to depend on what that actual particle or fluid is. Chloroplasts. Okay, chloroplasts are found in some, and these are going to be those that are going to be plant-like or plants themselves. Okay, so plants and algae, because they're going to be used for photosynthesis. Chloroplasts resemble mitochondria in the fact that they are double membrane bound organelles, and that they're going to have, instead of the cysti, they're going to have the thalkaloids, which are basically like little discs or stacks in there, and they're going to be, again, as I said, used for photosynthesis. And then there are going to be cytoskeletal structures. That is not a membrane-bound organelle, but the cytoskeletal structures are going to be made up of different protein structures known as actin, microtubules, intermittent filaments, etc. And they are going to be basically, if you will, the skeleton of a cell. So they're going to give it that internal scaffolding or structural support. Okay, so that is our basic overview of eukaryotic cell and their main functions of each of those different organelles or structures.